Hello, you are now listening to the first video lecture of the Human Anatomy and Physiology 2, our textbook, Human Anatomy and Physiology by Elaine Marieb and Katja Hohen, is available to you as an ebook under the Mastering AMP for students. Just go there, click on a module, and start viewing your ebook. Right now, you're listening and you are viewing the first video lecture for AMP2, and that is on the blood. The blood is a life-sustaining transport vehicle of the cardiovascular system. It is the river of life. It surges within us, transporting nearly everything that must be carried from one place to another. Now, long before the modern medicine, blood was viewed as a magical elixir that held the mystical force of life. Because it is drained from the body, when it is drained from the body, life would depart. We will all die if we lose the blood. Now, clinicians still regard it as a very important part of the body, and it is actually being examined now more often than any other tissue when we are trying to determine the cause of a disease in their patients. Now, in this chapter, we will describe the composition and function of this life-sustaining fluid that serves as a transport vehicle for the organs of the cardiovascular system. To get started, we need a brief overview of the blood circulation, and the blood itself is initiated by the pumping action of the heart. So it starts with the heart and then it goes through the arteries which branch repeatedly until they become tiny capillaries would still continue to carry the blood. And pretty much this is the way we serve, or the blood would be serving the even the remotest part of the body. Oxygen, nutrients, water, and everything else that leave the blood can cross through the capillary walls into the body tissues. Then carbon dioxide and waste from the body tissues can actually enter into the blood vessels to be carried by the blood back into their excretory organs or back into circulation. So blood is a transport system, but this is not the only function of the blood. It is for transport, it is also for regulation, and for protection. The transport function of the blood includes delivering oxygen and nutrients to the body, as I have mentioned, transporting metabolic waste to lungs and kidneys for elimination, transporting hormones from endocrine organs to the target organs. The regulation includes maintaining body temperature by absorbing and distributing heat, maintaining normal pH using buffers. It is also an alkaline reserve of bicarbonate ions, and it is actually maintaining the adequate fluid volume in the circulatory system. The blood is also being used for protection. It is preventing blood loss because of the things that you find inside a plasma. The plasma contains proteins and platelets in the blood, and they pretty much help in holding the blood inside the body. The blood also can initiate clot formation, which will prevent the blood from exiting just in case there's an accidental breakage of the blood vessel. So the blood is also preventing infection because there are agents of immunity that are present in the blood, such as antibodies, the complement system or the complement proteins, and white blood cells. So let's take a look at what you find in the blood, the composition of the blood. The blood is the only fluid tissue in the body. It's a type of connective tissue. It's not an organ. It is a connective tissue. It is made up of a matrix, and the matrix is a non-living fluid, which we call the plasma. And the cells in the blood are the living blood cells, which are called formed elements. And these cells are suspended in the plasma. And these formed elements are your erythrocytes, which we call, or otherwise known as the red blood cells, and also the leukocytes, otherwise known as the white blood cells, and the platelets. Now we can examine the blood, put it in a test tube, and spin it using a centrifuge. Spun blood would yield three layers. It would yield, it will actually divide into three layers, with the heaviest one sinking at the bottom and the lightest one would be on top. There is also a middle layer, which is actually the one that is not too heavy and not too light. The erythrocytes, or the red blood cell, would sink in the bottom. This is the heaviest component of the blood itself. When we say hematocrit, it is actually referring to the percentage of blood volume that is the red blood cells, the erythrocytes. And on the average, an average person's erythrocytes would be about 45% of his whole blood. The actual normal values would be 47% plus or minus 5 for males and 42% for females. The males would be kind of like, would have more red blood cells than the females because of the presence of a special male hormone, which called testosterone, that would stimulate the production of erythropoietin, which is the hormone that is stimulating the production of red blood cells in humans. The females has only 42% plus or minus 5. White blood cells would be less heavy. Both the white blood cells and the platelets would accumulate in the middle part of the hematocrit, forming what's called the buffy coat. The buffy coat accounts for 1% of the total blood volume. And it is actually very thin and it is whitish. It is between the red blood cell, the bottom part is your red blood cell, and then the top portion is actually the plasma layer. Now, 55% of this spun blood is actually the plasma, which is on top. And right here is the hematocrit itself, and showing you the major components of the whole blood in the hematocrit. When the blood has been collected and placed into a centrifuge, this is your centrifuge right here, and it will actually be divided into three parts. The bottom part, which is the heaviest part, which is made up of the erythrocytes, accounts for about 45% of the blood. Then you have the buffy coat, which is your white blood cell and your platelets, and then your plasma, which is 55% of the whole blood. It is the least dense component of whole blood. So, blood is sticky, opaque fluid with metallic taste. The color varies because of the oxygen content of the blood. When the oxygen content is high, as in the blood in the arteries, then the blood would be bright red. You call it scarlet red, I call it bright red. I'd say it is bright red. And then when it is low in oxygen, it would be slightly on the dark red and maroon red. The pH of the blood normally would be between 7.35 to 7.45 in the pH scale. It makes up about 8% of the total body weight. And the average volume in male is 5.6 liters, 5 to 6 liters, and the females will be 4 to 5 liters. Blood plasma is a straw-colored sticky fluid which is about 90% water. And it has about 100 dissolved solids that's made up of nutrients, gases, hormones, waste proteins, inorganic ions, plasma proteins, um, pretty much like uh, globulin and albumin and um, fibrinogen. These are the proteins in the blood. You could just imagine, because of the amount of protein, this is the one that pretty much creates an oncotic pressure and prevents blood from escaping out of the body. Now, table 17.1 in your textbook is a table that gives us the composition of the blood, gives a name of the substance and the description and importance of each of its constituents. This is actually something that you could work on and use whenever you are reviewing for your exam. Please make sure that you read on this, read on the constituents, and read on the functions of each of the constituents. Make sure that you understand it, make sure that you know them because they are going to be in the exam. All right, so formed elements are your red blood cells, your white blood cells, and your platelets. These are the cellular aspects of the blood. It's funny how they call themselves cellular aspect, or they're actually, well, during my time, we call it cellular aspects. Now we don't call it anymore cellular aspects. Look at not everybody or not everything that is in floating in the plasma are actual cells. Some of them are only fragments of a cell. Some of them are not complete cells. As a matter of fact, only one particular element is a complete cell, and this will be your white blood cell. Now here is your blood cells. And this is the electron micrograph of your blood cell. And you could see that you have the erythrocytes. These are your red blood cells. Notice that your erythrocytes here does not show any nucleus. That's why they are not complete cells, but they are cells. Then you have the platelets right here. Your platelets are really fragments only of a megakaryocyte, a very large cell that during the process of development exploded. And that's actually the fragments themselves, and they call them the platelets. The leukocytes, these are the true cells. You open this and you have a nucleus in there, you have the endoplasmic reticulum. It's a complete cell by itself. So this is a real blood smear. The blood is actually smeared on top of a not a test tube but a histology slide, and they actually immersed it in a stain. And if I'm not mistaken, the stain that was used here is right stain. And you could see, if you stain blood with right stain, that the complete cells would actually show their nucleus and some of the things that are inside the cell, like granules, okay? So if you see granules in your white blood cell, those are called granulocytes. If you don't see any granules, like this one here is a very small one with no granules in the cytoplasm, that is the agranulocytes. And we are going to get into this. And these cells here, the pink cells that does not show any nucleus, these are your red blood cells. So let's now take a look at the erythrocytes. The erythrocytes, or red blood cells, are small cells, about 7.5 micron in diameter, and they are shaped like biconcave discs. They are anucleated and essentially has no organelles. They're filled with hemoglobin for gas transport, and the red blood cell diameters are larger than the capillaries. Some contain uh, membrane protein, which we call spectrin, and other proteins. Spectrin provides flexibility to the cell that enable it to change shape. A very good example of complementarity of structure and function. There are three features that make up for efficient gas transport of the red blood cell. First of all, it is biconcave. Pretty much because it is biconcave, it is nearly flat. It offers a huge surface area relative to volume for gas exchange. Pretty much like everything that is inside the cell could be or could actually be in direct touch with its environment. Hemoglobin makes up 90% or 97% of the cell volume, not counting the water part, and the red blood cells do not have any mitochondria. Imagine that. If you are a red blood cell and you will have a mitochondria, then you are going to be inefficient in your job because you are supposed to be carrying oxygen. And what does mitochondria do? Mitochondria produces ATP, and for a mitochondria to produce ATP, it would need to consume oxygen. So your red blood cell do not have any mitochondria, because if it will have mitochondria, it's not going to be efficient in its function to carry oxygen. Most of the oxygen that it will carry, it will consume by itself. All right? So it doesn't have any mitochondria. Just make sure that you remember that. And here's exactly how your red blood cell would look like. It is biconcave. It doesn't have any nucleus, the mature one. Although the immature red blood cell would have the nucleus. Because how do you expect the red blood cell to become a red blood cell, a mature red blood cell, if it will not start from something? Of course, it started from a baby erythrocyte, and it became a mature erythrocyte with the help of the nucleus. Remember that your nucleus is the control center of the cell. It directs the development of a cell. So before you lose the nucleus, or before a red blood cell will lose the nucleus, it would have to first do all of the job that is needed for it to become a perfect red blood cell or adult red blood cell. Then it would lose the nucleus. So your erythrocytes would need to be dedicated for gas transport. There is a special molecule that you find inside your red blood cell, and you call this the hemoglobin. The hemoglobin binds reversibly with oxygen. It means it can bind into the oxygen, and then it can also let go of the oxygen. The normal values in hemoglobin the normal hemoglobin values in a cell would be 13 to 18 grams per 100 ml for males, and for females it's 12 to 16 grams per 100 ml of blood. The hemoglobin inside the red blood cell consists of the red hemipigment that is bound to a protein globin. And the globin is actually made up of 4 polypeptide chains, where 2 would be an alpha polypeptide and 2 would be beta chains. The hemipigment is bonded to each globin chain, and this is the one that gives the hemoglobin, or the blood itself, its red color. Because the heme in the central portion of the heme is where you will find the iron molecule that can bind one oxygen. The iron molecule, once oxygen is bound to it, will turn bright red. From a dull red to a bright red. From a dull red, which is almost like brownish red, to a very bright red once oxygen gets into it. So this is your hemoglobin. It has 4 globin chains. This is your beta globin chains. This is the alpha globin chains, and there are hemes on each of the globin chains. So you have 4 hemes. Each heme is capable of carrying 1 oxygen molecule. Now take a look at this. You have 4 hemes, and therefore And a hemoglobin is actually carrying 4 oxygen molecules total. And one red blood cell is actually 97% hemoglobin. It does not have much inside it except pure hemoglobin, because its only function really is to carry the oxygen, and therefore it needs to be as effective as possible. It cannot have any other form elements such as your mitochondria or whatever. It has only hemoglobin. So it is really true that it is successful in the transport, but how long do you think would a cell live if it doesn't have any nucleus? If it doesn't have any of the other formed elements inside it that will help it survive for a long time? Not that much. See, the lifespan of a red blood cell is not very long, whereas your white blood cell could live sometimes for the length of the life of the person. Your red blood cell is only going to last less, in months only. There you go. That is your hemoglobin. So when oxygen gets loaded in the lungs and would come in contact with hemoglobin, it will produce a complex molecule, which you call oxyhemoglobin. Oxyhemoglobin is the one that turns bright red or ruby red. When oxygen is unloaded in the tissues, and you will have a hemoglobin that would probably have only like 1 oxygen in it left, or 2 oxygen left, which means 2 of the oxygen has pretty much like left the hemoglobin. It will produce another molecule or another complex molecule, which you call deoxyhemoglobin or reduced hemoglobin. And the color of the reduced hemoglobin is dark red or brownish red. Carbon dioxide loading is also loaded in the tissues. 20% of the carbon dioxide in the blood binds to hemoglobin, producing a complex molecule, which you call carbaminohemoglobin. Carbon dioxide doesn't have good affinity with oxygen, like that's not good affinity with hemoglobin like oxygen does. Oxygen has extremely good affinity, very strong affinity with hemoglobin. So between hemoglobin, when hemoglobin actually comes in contact with both carbon dioxide and oxygen, it will definitely be picking up the oxygen and would leave the carbon dioxide, but um, would leave the carbon dioxide. So what happens to the carbon dioxide? Well, the carbon dioxide, never mind if it doesn't have much of a carrier, or never mind if the hemoglobin doesn't want to carry it, the carbon dioxide is soluble enough in the water that it can actually travel throughout the entire body with the help of just the plasma. Most of the carbon dioxide is dissolved in the plasma. So let's take a good look at the lifespan, or maybe the hemopoiesis, or the production of or formation of all blood cells. Hemo means blood, and poiesis is production of. These occurs in the bone marrow, composed of reticular connective tissues and blood sinusoids. If you recall your um, bone tissue, in the bone tissue there is actually your compact bone, and there's also your spongy bone. In the spongy bone you will find trabeculae, and inside the pores of the trabeculae you will find the red. You call this the red bone marrow simply because we call it red bone marrow, because most of the components that makes it slightly red is actually the red blood cells that is being produced there. In adult, they're found in axial skeleton, the girdles, the proximal epiphysis of the humerus and the femur, and the pelvic bones, the girdles, I meant. So the hemopoietic stem cells, the great grandmother of all the blood cells, is what you call the hemocytoblast. There's only one great grandmama of all the blood cells, the hemocytoblast. From this one cell would pretty much like emanate many different kinds of cells. You have the erythrocytes, so the red blood cells, and you have all of the white blood cells. How many different types of white blood cells do we know? Well, we have 5 different types of white blood cells, plus your erythrocytes. Whoa, that's really a lot of cells coming only from one great grandma. How is it that one cell can actually become 5 different cells or 6 different cells? Well, it depends on how we stimulate it. There are different stimulating factors and proteins that would pretty much like that would make it become a different cell. And these different factors or these different hormones would provide the information on what it is going to be. If it receives that information to become a red blood cell, it becomes a red blood cell. If one of them receives information for it to become, let's say, a basophil, it will become a basophil. Or if there's going to be an information for it to become a megakaryocyte so that there will be platelets, it will become megakaryocyte. I beg your pardon, it's not only for 6 blood tests, but blood cells, but it's actually, I forgot, there's actually also the platelets, 7. So we call it the stem cell, and it gives rise to all formed elements. Hormones and growth factors push these cells towards a specific pathway of blood cell development. When the cells become committed to become whatever it is supposed to be, then it cannot go back to become a hemocytoblast. It cannot change back. The new blood cells will have to move forward in developing into an adult. So let's take a look at the first development of the first formed element that we're going to take up today, and that is the erythrocytes. We call the development of erythrocytes erythropoiesis. It is a process of formation of red blood cells that takes about 15 days in the bone marrow. And there are several stages of transformations. First of all, guys, the first stage is when your hemopoietic stem cell, this is your great grandmother, the hemocytoblast, would transform into a proerythroblast. That is your myeloid stem cell pretty much transforming or becoming a proerythroblast. Now, this proerythroblast is already a committed cell. Once committed, it cannot anymore turn back to become a hemopoietic stem cell. It'll have to go forward. From a proerythroblast, this proerythroblast will divide many times, transforming into a basophilic erythroblast. Now, in the process of division, it develops. This basophilic erythroblast would produce a lot of ribosomes. Now, what is that cell component supposedly doing? What is ribosome doing inside the cell? Ribosomes are the ones that is engaged in protein synthesis. So it seems that this particular ribosome is actually synthesizing protein. And I guess a protein that is actually being synthesized here is your hemoglobin. So your your basophilic erythroblast will now become a polychromatic erythroblast. This type of erythroblast now would be changing a little bit in color. It is now because of the accumulation of hemoglobin inside it. So hemoglobin continues to accumulate, when there is enough hemoglobin inside the cell, then the function of the nucleus is already done. The nucleus now can already be ejected. So the nucleus has been ejected in this area here, and now you have a baby red blood cell that is ready to be ejected into the bloodstream. This baby red blood cell do not have any nucleus, but would not show any concavity yet. But you have a good percentage. 1.5% of the blood is actually reticulocyte. Every day, we lose 1.5% of our red blood cells, and every day our bone marrow will replace it with 1.5%. So you will see in a blood count, there will always be 1.5% of this baby red blood cell, which you call a reticulocyte. Because this is supposed to be a standard count, 1.5% of our blood is reticulocyte or baby red blood cells. When you see a blood smear that would have a count of more than 1.5% reticulocyte, that means that the bone marrow of the person is producing a lot of red blood cell. If the red blood cell becomes 4%, that is really a big change. You are normally supposed to form only 1.5% of your blood. How come your blood is 4% reticulocyte? Something must be wrong. Well, the doctor would firmly conclude that maybe the person is losing blood. That's why he is actually producing that much blood. Whatever your body is doing is always a response to something. So when you're losing blood, your body will produce more blood to compensate for the loss. So if your reticulocyte count is 4%, that means you are producing 4% blood, which is actually like a lot more than the normal, because the normal is only 1.5%. Now the doctor will have to find out what is it that you are, where is it that you are losing blood? Why are you losing blood? So this becomes now a significant point of as screening. When the doctor wants to find out what's wrong with you, one of the things that is done is actually the blood cell. And for the red blood cells, the reticulocyte count and the hemoglobin concentration of the red blood cell is significantly going to give us the chance to find out if there's anything wrong with all of you. So we're done with this. Let's now go into the regulation and requirements of erythropoiesis. Okay, so you are producing red blood cells. Inside these red blood cells, you would need the things that you need to form hemoglobin. Okay, so if you are not going to have all the raw materials that is needed to produce the hemoglobin, then your red blood cells will either be too small because it doesn't have much hemoglobin in it, or your red blood cells will be less in number. What are the consequences of this? Well, too few red blood cells would lead to tissue hypoxia, but too many red blood cells would increase blood viscosity. Approximately more than 2 million red blood cells are made per second, and that's a lot. There's a balance between red blood cell production and destruction depending on the hormonal control as well as dietary requirements. So the formation of red blood cell pretty much is tantamount to how the hormones are controlling the formation and the dietary requirements. So we need something to control the start of the erythropoiesis, and we need something to make it continue. We need the stuff, the nutritional things that we need in order that the red blood cell will become a full red blood cell. And for the most part, the one that we need here is a protein to form the hemoglobin. So let's take a look at the hormonal control. The hormone that would stimulate the production of red blood cell is what you call erythropoietin, EPO. There's always a small amount of EPO in the blood to maintain the basal stimulating rate, so that every day you are producing 1.5% of your red blood cells. It is released by the kidneys, and there are some released by the liver in response to hypoxia. Hypoxia means loss of oxygen. If your body can feel that your oxygen in the blood is low, then you are going to be producing erythropoietin in the kidneys or in a little bit from the liver. This is actually at low oxygen levels because there are oxygen sensitive enzymes in the kidney cells that cannot degrade the hypoxia inducible factor. Pretty much anytime there is actually a decrease in the oxygen level of the blood, the liver of the kidney will always sense it and would start production of EPO. So what are the causes of hypoxia? Well, hemorrhage is one of them, or increased destruction of red blood cell. Anything that will result in the level or the number of red blood cell going down could lead to hypoxia. Or if there are insufficient hemoglobin per red blood cell, which means your hemoglobin, the hemoglobin content of red blood cell is too little, then you are going to have smaller red blood cell. And that would pretty much be, this particular character is found in iron deficiency anemia, or reduced availability of oxygen. And that is happening whenever we are in high altitudes, or if we have some lung problems such as pneumonia. Now, too many erythrocytes or high oxygen levels in the blood would inhibit EPO production, which means that if your erythrocyte is already high, then your body will not be producing and will not anymore be producing too much erythrocyte. It doesn't need to. If it has a lot of it, it doesn't need to do anything anymore. Or if your oxygen is high enough, then your bone marrow will stop producing erythrocytes. Now the EPO or erythropoietin would cause the erythrocyte to mature faster. The hormone testosterone of males would enhance EPO production. That's why the males have higher red blood cell count. It's because of their testosterone. Now, this is the homeostasis for the EPO mechanism for regulating erythropoiesis. First of all, guys, on this end here, your stimulus is hypoxia, meaning low oxygen level or inadequate oxygen delivery because of decreased red blood cell count, decreased amount of hemoglobin, decreased availability of oxygen. It moves into the kidney, and then the stimulus or the information is taken in by the kidney or the liver in smaller extent, and the kidney will start to release erythropoietin. Erythropoietin now is received by the bone marrow, and will stimulate the bone marrow in its production of red blood cell. There you go. Red blood cell now that's been produced in the bone marrow, after 15 days is going to be released in the bloodstream. And immediately after it's released in the bloodstream, then it will it will reverse the initial stimulus, which is actually hypoxia. So now, after red blood cells are already released in the blood, then the oxygen carrying ability of the blood will rise and the hypoxia is gone. Let's take a look at some clinical imbalance in red blood cell production. There are athletes who would abuse the use of artificial erythropoietin. The use of EPO would increase hematocrit or increase red blood cell count, which would allow the athlete to increase stamina and performance. How would the athletes do this? Well, there are different ways of increasing the hematocrit. One of them is what you call blood doping, meaning before like a week before a particular competition, the or a month before a particular competition, the athlete would already be producing enough red blood cell and would pretty much put the red blood cell in a bank, and then a couple of days the before the competition, he will get some of the blood so that his red blood cell count or his hematocrit will rise, and that will increase his ability to fight because he will have a lot of oxygen. Remember that when you are using your muscles, you are using a lot of oxygen. And so, whenever you have a lot of oxygen, then you are going to be strong. Your muscles are always able to contract because of the oxygen around. But the dangerous consequence is that the EPO can increase hematocrit from 45 or even 65% with dehydration, concentrating the blood even more. The blood becomes like a sludge and can cause clotting, stroke, or heart failure. You put in more blood into yourself because you want to have more oxygen. You are actually going to turn your blood into a sludge, and you are going to increase your chances of stroke because of the blood clot that's going to float around, or even heart failure. So let's take a look at the regulation and requirements of erythropoiesis. So, because we are producing proteins, we will need the amino acids, the lipids, and the carbohydrates. We will need also, because we are producing hemoglobin, we will need the iron, so which is available in the diet. 65% of the iron is found in hemoglobin, with the rest being in the spleen, the liver, and the bone marrow. Free iron are toxic, so iron is always bound with proteins. The iron that is stored in the cell is stored as ferritin and hemosiderin. And when it is not stored in the cells, it is transported in the blood. And so the iron, so you cannot have free iron in the blood, is now going to be bound to a protein which we call transferrin. There is never a chance that the iron will be free iron that's floating around or within the cell. It is always bound to a protein. Vitamin B12 and folic acid is necessary for DNA synthesis for rapidly dividing cells, such as developing red blood cells. So, B12 and folic acid are necessary in the production of red blood cells. The lifespan of your red blood cell is about 3 months. So every 3 months, a newly born red blood cell will live only 3 months. After the third month, it will go to the spleen, which is the graveyard of red blood cell, and there it will die. They are anucleated, so they cannot synthesize their new proteins or grow or divide. And all red blood cells become fragile. The hemoglobin begins to degenerate and can get trapped in small circulatory channels, especially in the spleen. And I mentioned to you they are the graveyard of the red blood cells. And a macrophage in spleen engulfs and breaks down the dying and overly sized red blood cells. So when the red blood cell breaks down, It breaks down into its heme component, into its iron component, and the globin component. They're all three are actually separated. The iron binds to ferritin and hemosiderin and is stored for reuse. So we are actually recycling the iron. We don't lose the iron. But is that true? Don't we lose iron? Well, the only time you're going to be losing iron is when you lose whole blood. Women are more generally inclined into iron deficiency anemia because we are having our monthly period. When you are having your monthly period, you are actually losing a lot of blood. And you lose that blood, and together with the iron that is in the blood, you lose it. So we have to replenish the iron that we have lost when we are having our period. The men will not ever have any worries about having a period because they don't have a period. But the thing is, the only time a man would probably have iron deficiency anemia is when he has either a bleeding ulcer or anything, any disorder that, or maybe he had um, um, surgery and is actually bleeding, or had what do you call this? He was stabbed or whatever, and is having hemorrhage or what. But that's the only time a man would pretty much like experience uh, iron deficiency anemia because of loss of blood. Uh, Normally, under normal circumstances, a man would not have iron deficiency anemia. When red blood cells are degraded, the iron and the heme splits. The iron is bound to hemosiderin and ferritin. The heme is degraded into yellow pigment which you call bilirubin. The liver secretes bilirubin into the intestine where it is degraded into urobilinogen and is transformed into a brown pigment which you call stercobilin. The stercobilin pretty much leaves the body in the feces, and pretty much it is actually the one that makes your feces yellowish. If you eat a lot of fats or food that is rich in fat, then the bile, which carries stercobilin in it, the bile will be secreting a lot of this stercobilin, and will actually be together with the food that you are digesting that is rich in fat, and therefore it turns your feces yellowish. So you will know if you are really eating food that is high in fat. Your feces is yellow, and that's not really healthy. A healthy feces would be one that would have the color of a digested vegetable, and that will be brownish, not yellow, never yellow. And here you are, guys. This is the life cycle of your red blood cell. Okay. I am going to leave this to you. I have already started this. This will tell you exactly what it is. The new, you're showing the kidney here because the kidney is the one that is producing erythropoietin. Erythropoietin levels in the blood is released. Erythropoietin is released into the blood by the kidney. That will stimulate the production of red blood cell by the bone marrow. That's your bone marrow right there. Okay. The bone marrow now will be releasing red blood cell into the bloodstream. And then once it is already old, very old red blood cell, it will now break down. The hemoglobin breaks down into the heme component and the globin component. The heme component is broken down into bilirubin and iron. The bilirubin is picked out by the liver and would eventually be secreted into the intestine when we are digesting fat. The heme part, okay, is actually stored as ferritin or hemosiderin in the blood, and iron is actually transferred back into the blood and could be reused in the production of new blood cells. So, the food that we eat, the nutrients, amino acids, iron, B12, folic acid, these are all absorbed from the intestine and they enter into the bloodstream. These are all necessary in the production of red blood cells. So let's take a look at the disorders of erythrocytes. Anemia is actually one of the disorders, and polycythemia is the other one. Anemia is when the blood has abnormally low oxygen carrying capacity, that is too low to support normal metabolism. It is a sign of a problem rather than a disease itself. Symptoms of anemia would be fatigue, pallor, dyspnea, and chills. Three groups are based on cause. Blood loss can cause anemia. Inadequate production of red blood cell can cause anemia. Too many red blood cells being destroyed can also cause anemia. Anemia brought by blood loss is what you call hemorrhagic anemia. This is brought about either by rapid blood loss, such as a gaping wound, and is treated by blood replacement. Chronic hemorrhagic anemia. This is slight but persistent blood loss. An example of this is when a person has hemorrhoids, or bleeding ulcer. Primarily, the problem must be treated in order to stop the blood loss. Let us take a look at the types and relative percentages of leukocytes in normal blood. In a spun blood, the erythrocytes are at the bottom. That's what the hematocrit is. It accounts for the hematocrit. The buffy coat will be your leukocytes and your platelets together. This buffy coat here. And above this will be the plasma. The leukocytes is about 4 to 8,000 to 10,800 microliter per microliter of blood. And you have granulocytes and agranulocytes in that level, in that number right there. 70%, 50 to 70% of that will be neutrophil. Eosinophil will account for 2 to 4%. Basophil is about 0.5 to 1.5%. The lymphocytes account for about 25 to 45%. The monocytes will be about 3 to 8%. These are your leukocytes. This is the neutrophil. You will have to know exactly what a neutrophil would look like. In the lab exam, you will have to identify the neutrophil in a microscopic slide. This is your basophil. It would usually have two lobes. It would have red granules. You could see the red cytoplasmic granules, whereas the granules of the neutrophil will be slightly pink. The basophil would have red. And the granules, can you see those dark blue granules? That's actually the granules of a basophil. The lymphocytes are small and they are agranulocyte and they don't have any cytoplasmic granule. You could see it. And your monocytes are the biggest of all 5 white blood cells. They have kidney-shaped nucleus or C-shaped nucleus. They have abundantly pale blue cytoplasm without any granules. So your larger and short-lived red blood cells are granulocytes. They contain lobed rather than circular nuclei. Their cytoplasmic granule was stained specifically with right stain, and they are all phagocytic to some degree. This will be your neutrophil, your eosinophil, and basophil. Your neutrophils, also known as the polymorphonuclear or the polys, because the nucleus is lobular, pretty much account for they they kill microbes and in a process which you call the respiratory bursts. They are phagocytic. They are referred to as the bacteria slayers. They have defensive granules that merge with the phagosome. They form spears that pierce holes in the membranes of ingested microbes. Eosinophils. The nucleus are bilobe. They play a major role in allergy and in asthma, as well as immune response modulators. Basophils. They are functionally similar to mast cells. They secrete histamine, which is an inflammatory chemical that acts as a vasodilator and attracts white blood cells to the inflamed sites. Among the agranulocytes, you have the lymphocytes and the monocytes. Both of them have kidney-shaped nuclei, but the kidney shape, almost like C shape, belongs to the monocyte and it's very pronounced. The lymphocyte is the smallest. It is the second most numerous white blood cell. It accounts for about 25% of all white blood cells. And there are 2 types of lymphocytes. Oh, well, lymphocytes hide in the lymph nodes. You find them in lymphoid tissue. The lymph nodes, the spleen, the tonsils. Very few of them circulate in the blood, not unless they need to. They're crucial to immunity. And there are 2 types of lymphocytes. When we get into the immune response, I think that's chapter 20 or 21, we are going to pretty much like discuss what the actions or the functions are of the lymphocyte. There are 2 types of lymphocytes. Lymphocyte T, or otherwise known as the T cells. They act against virus-infected cells and tumor cells. And the B lymphocytes or B cells, they give rise to plasma cells, which produce the antibodies. Monocytes, largest of all leukocytes, and they account for 3 to 8% of all white blood cells. They activate lymphocytes to mount an immune response. They are actively phagocytic cell. They are crucial against viruses. They are intracellular bacterial parasites and they proliferate when there is chronic infection and intracellular bacterial parasite. Let's take a look at the production and lifespan of leukocytes. Leukopoiesis, they all emerge from one great-great-grandmother, and that is your hemocytoblast. The great hemocytoblast, which is responsible for producing the red blood cell, is also the great-grandma of all the white blood cells. The production of white blood cell is what you call leukopoiesis. And instead of your erythropoietin with the erythrocytes, the leukocytes would need interleukins and colony stimulating factors. These are the ones that stimulates the production or the formation of the different types of white blood cells. All of them originate from the hemocytoblast. This is the stem cell that branches into two pathways, the myeloid stem cell and the lymphoid stem cell. This lymphoid stem cell produces lymphocytes. You could see there at the bottom, you have your B lymphocyte and your T lymphocyte. They both emerge from the lymphoid stem cell. The myeloid stem cell here produce all other elements, the eosinophil, the basophil, the neutrophil. These are all granular white blood cell. And then you have one agranular white blood cell that is also coming from the myeloid stem cell. And that will be your monocytes. The monocytes pretty much where is actually the cell where your macrophage tissues would emerge also. They become the macrophages, the phagocytes. So, guys, this is really very automatic. I'm not anymore going to be discussing the production of the lifespan of leukocytes. Just make sure that you know the myeloid stem cell will produce the three granular white blood cell and the monocyte, and the rest of the agranulocytes, your lymphocytes, is coming from the myeloid stem cell. I am not anymore going to discuss the stepwise thing, and I am not going to ask you any question in the exam on that. So let's take a look at some homeostatic imbalances. Now, many hemopoietic hormones, the EPO, and the colony stimulating factors are used clinically to induce or stimulate the bone marrow of cancer patients receiving chemotherapy or stem cell transplant. They are used in pretty much like stimulating the slow, sluggish bone marrow of cancer patients because their bone marrow are actually being killed by the chemotherapy. So these hormones, the EPO and the factors that stimulate the production of white blood cells, is being used in order to revive the sluggish, cancer-laden cells, hemopoietic cells of cancer patients. They're also used to increase protective immune responses of AIDS patients. Overproduction of abnormal white blood cell will give rise to leukemias and infectious mononucleosis. An abnormally low white blood cell count is what you call leukopenia. This could be drug-induced, particularly by anti-cancer drugs or glucocorticoids. Another disorder is the leukemia. Leukemias. These are cancerous conditions involving the production of abnormal white blood cell. Usually involve clones of single abnormal cell. They are named according to abnormal white blood cell clone involved. Myeloid leukemia involves myeloblast descendants. lymphocytic leukemia involves lymphocytes. These leukemias are acute. They could be acute or they could be chronic. Acute would be quickly advancing. Chronic will be slowly advancing. Acute leukemias derive from stem cells. They primarily affect children. The chronic would be primarily affecting older people. This leukemia involves proliferation of later cell stages. Without treatment, these leukemias are fatal. The immature and non-functional white blood cell would flood the bloodstream. The cancerous cells fill the red bone marrow, crowding out the other cell lines, and then they can lead to anemia and bleeding. Death is usually from internal hemorrhage or overwhelming infections. Now let's take a look at the platelets. Platelets are fragments of larger megakaryocytes. They stain blue or purple and they're actually present together with the rest of the blood smear. This is what a platelet would look like in a blood smear, but they come from an abnormally large cell, which you call a megakaryocyte. This type of cell will divide inside without cytoplasmic division. All the nucleus will actually divide until it has already like reached something like 6 or 8 cell, 8 nuclear stage, and it becomes so big that it cannot anymore hold what is inside, and they will burst. And then all of the other fragments that would be resulting from the bursting will be what you call the platelets. Circulating platelets are kept inactive. When they're activated, they're dangerous. They are going to cause the blood to clot. They're kept inactive by mobile nitric oxide and prostacyclin from the endothelial cells that are lining the blood vessels. It is necessary that we have this nitric oxide, and prostacyclin, so that the platelets um, will stay inactive. The chemicals that are involved in the clotting process is serotonin, calcium, also a group of enzyme, ADP, your PDGF, the platelet-derived growth factor. All of these are involved in the production of platelet clot. The production of platelet is regulated by, again, a hormone which you call thrombopoietin. It is formed in a myeloid line from a megakaryoblast, the stage 1 megakaryocyte. There's actually several mitotic incident that will be coming in that will be resulting in the enlargement of the cell. And once it becomes multi-lobe nucleus and it cannot anymore hold to the size, it will burst. A normal platelet count will be about 150,000 to 400,000 platelets per ml of blood. Guys, I put this table here. It is in your textbook. This is a good cheat sheet, or it's a good sheet for you to study your different blood cells. Okay, let's take a look at now we are in a hemostasis. Hemostasis is a fast series of reactions for stoppage of bleeding. They require clotting factors and substances released by platelets and injured tissues. And there are 3 steps in hemostasis: vascular spasm, platelet plug formation, and coagulation or blood clotting. Vascular spasm. This is triggered by direct injury to vascular smooth muscle. It is also triggered by chemicals released by the endothelial cells and platelets. And it's also triggered by pain reflexes. They're most effective, these vascular spasms is actually mostly occurring in smaller blood vessels. They can significantly reduce blood flow, and then all other organs or mechanisms can actually kick in. So what is spasm? This is actually the part where after you have a um, an injury and there's already blood that is actually oozing out of the injured or the cut blood vessel, this blood vessel would need to close up, but it's not really just going to close up. So before before it closes up, the body needs to do something to hold the bleeding, and vascular spasm is actually the first one. Then the next one is what you call platelet plug formation. While the blood vessels are holding on tight not to allow blood to go through that cut end, platelets will now come into play. It will become activation. They stick to collagen fibers that are exposed when these vessels are damaged. Pretty much the platelets do not, in all seriousness, platelets do not get activated. They do not stick to the intact blood vessel wall. Once collagen is exposed, once there is a cut and collagen is exposed, then your platelets will become activated and will go for platelet plug formation. There are also prostacyclins and nitric oxide secreted by the endothelial cells. These are also preventing the platelets from sticking and from reacting. When activated, the platelets will swell. They become spike and sticky, and they release chemical messengers such as ADP, which cause more platelets to stick and release their contents to each other. So what happens is that the platelets now, imagine, if they are sticky, they adhere to one another. They stick very close to one each other. They clump. Then serotonin and thromboxane would enhance more the vascular spasm that has already been started by the cut and more of platelet aggregation. This is actually what you call a positive feedback mechanism. As more platelets stick, they release more chemicals, which give more platelets, which causes actually more platelets to stick and release more chemicals. Platelet plugs are fine for small vessel tears, but larger vessel breaks will need an additional step. So for the smaller skin abrasions and cuts, your platelet plug will work. But for big tears that will have, for big tears in big blood vessels, platelet plug is not going to work. After the platelet plug, we are assuming that we are actually just dealing with the regular cut, skin cut. Coagulation will follow. Coagulation is blood clotting. It will reinforce platelet plug with fibrin threads. The blood clots are effective in sealing larger vessel breaks. So this coagulation will help in sealing cuts that are originating from larger vessel breaks. Blood here during coagulation is transformed from the liquid blood to a gel-like clot. A series of reactions of clotting factors, which we call procoagulants, these are mostly plasma proteins, will trigger extrinsic and intrinsic reactions. These plasma proteins or clotting factors are numbered 1 through 8 in the order of discovery, and vitamin K is needed to synthesize 4 of this. And coagulation occurs in 3 phases. Right here, we have the 3 phases. You have the vascular spasm, which we have discussed. platelet plug formation, which we have discussed, and the last is actually coagulation. We are now going to get into coagulation. This is coagulation, which is made up of the intrinsic as well as the extrinsic pathway. These are initiated by either intrinsic or extrinsic and both. They are triggered by tissue damaging events. They involve a series of procoagulants, and each pathway cascades towards and ends with an activation of factor X and factor 5 to form a prothrombin activator. So in both cases, whether you're going by intrinsic pathway or you're going by the extrinsic pathway, both of them would have the same um, consequence, and that is the formation of prothrombin activator. I am not going to ask you in your exam what this pathway is. Could you imagine how many? Okay, the 7, the 12 becomes 12A, becomes the 11, becomes 11A. That is ridiculous. Okay, I am not going to ask you to be, because this is not biochemistry anyway. So just note that there are two pathways that is happening. The intrinsic pathway is brought about by vessel endothelium, which ruptures, thus exposing the underlying tissues. The collagen is the one that's very important. The extrinsic pathway, this is tissue cell trauma exposed blood to the tissue factor itself. So both of them though, both pathways though, will end up in the formation of prothrombin activator. There you go. Intrinsic means within, because the clotting factors are present within the blood. Extrinsic factor is called extrinsic because the factors needed for clotting are located outside the blood. They are in the tissues. They're triggered by exposure to tissue factor, also called the factor 3. They bypass several steps of intrinsic pathway, so they are faster. This is faster. This is a little bit slower, but both of them will come to the same specific the same specific factor, the prothrombin activator. They produce that. Now, when the prothrombin is already present, there is your prothrombin. The prothrombin activator catalyzes the formation of prothrombin to activate enzyme thrombin. And here you go. Let me go back. Doesn't want to go back. Anyway, so what causes the production of the clot itself? The clot itself is fibrin. The clot is an insoluble polymer. It is a fiber. How is fibrin going to come about? Fibrin comes from the protein fibrinogen. Fibrinogen is actually a soluble fiber. This is the fiber of the blood. The blood is a connective tissue, and all tissues have fiber, and the fiber of the blood is your fibrinogen, which is a protein in the blood. You don't see it as fiber, but once the blood clot starts, that particular fibrinogen gets converted into an insoluble form, which you call fibrin. This is an insoluble polymer, and it turns into cross-linked fibrin mesh. This is your blood clot. So this is your red blood cell that has undergone coagulation. So this is an electron micrograph of erythrocytes that is trapped in a fibrin mesh. So now you have your fibrinogen that's being converted into fibrin or blood clot. Okay, your clot does not just stay there just like the way it is. It must be stabilized for tissues to really like heal and reform. You have to make sure that that particular cut would be stabilized and blood will not move and go through there. So the while the white blood cells are working around and the other cells are working around rejuvenating the destroyed part, the clot must be stabilized and then they will be removed when damage has been repaired. But while the repair process is happening, the clot must be stabilized. And after that, uh, there's going to be fibrinolysis. Now, first clot retraction will have to happen. Clot retraction happens when actin and myosin in the platelet contract within 30 to 60 minutes after the coagulation starts. The contraction pulls on fibrin strands, squeezing serum from clot, and then draws the ruptured blood vessel edges together. So it actually pulls together the walls of the two uh, damaged edges so that they become close to one another. Thus, it will enhance the repair process. The vessel is healing even as the clot retraction will occur. Platelet-derived growth factor is released by the platelets. It stimulates the division of smooth muscles and fibroblasts to rebuild blood vessel wall. The vascular endothelial growth factor stimulates endothelial cells to multiply and restore the endothelial lining. Then fibrinolysis happens. This is a process where clots are removed after repair is completed. Granting that the repair has already been done, all of those clotted areas of that's a lot of clots will have to be removed. They cannot stay there. Plasminogen, which is a plasma protein that is trapped in a clot, is converted to plasmin. And plasmin is a fibrin digesting enzyme. Plasmin will digest all of those fibrin fibers, enhancing more or giving more space for more repair to happen. So, what are the factors limiting normal clot growth? There are two mechanisms that limit clot size. Number one, the swift removal and dilution of clotting factors and inhibition of activated clotting factors. There's a limited amount of thrombin, that is restricted to clot by fibrin. It's fibrin threads. Limited amount of thrombin is restricted to clot by fibrin threads, preventing clot from getting too big or escaping into the bloodstream. You have an antithrombin, but is antithrombin 3. This inactivates any unbound thrombin that escapes into the bloodstream. Heparin in basophil and mast cells inhibits thrombin by enhancing antithrombin 3. Why? What is thrombin? Well, thrombin is actually the one that promotes clot. So you want to inhibit or limit the amount of thrombin in order that the clot is going to be limited. You're finished already. The clotting process should stop. And so you should stop thrombin. Factors preventing undesirable clotting. Factors preventing platelet adhesion. Well, your platelets should go back to its original smooth size. They should not be active anymore. They should rest. So your smooth endothelium, the smooth endothelium of blood vessel would prevent the platelets from clinging to each other. The endothelial cells secrete antithrombic substance such as nitric oxide and prostacyclin. This prevents the platelets from sticking to one another. Vitamin E, quinone, formed when vitamin E reacts with oxygen, is a potent anticoagulant. That's the reason why those people who are on vitamin E regimen are being requested to stop their vitamin E about a week before surgery because vitamin E makes them bleed. Because vitamin E quinone, that is formed when vitamin E reacts with oxygen, is an anticoagulant. It prevents your blood from clotting. You have two other major types of disorders. Thromboembolic disorder, they result in undesirable clot formation. Bleeding disorder, which is abnormalities that prevent normal clot formation. Then you have your disseminated intravascular coagulation that involves the two types of disorders. So your thromboembolic condition, these are your thrombi and emboli. A thrombus is a clot that develops and would persist in an unbroken stem or unbroken blood vessel. These may circulate in the blood. They block circulation. They may lead to tissue death. An embolus is a thrombus freely floating in the bloodstream. This can cause what you call embolism. Embolism is an embolus obstructing a vessel. An example will be your pulmonary or cerebral emboli. The risk factors, these are risk factors of atherosclerosis, risk factors of inflammation, and slowly flowing blood or blood stasis from immobility. That's why people who are immobile and who are not really moving normally are very prone to embolism. So, what do we use? Well, we have anticoagulant drugs that is used to prevent undesirable clotting. Aspirin, heparin, warfarin, and pretty much I would say I would like to add here vitamin K, vitamin E rather, not vitamin K, vitamin E, because vitamin E is an anticoagulant drug. Thrombocytopenia, deficient number of circulating platelets. When platelet number goes down, then you have what you call thrombocytopenia. When platelet count is below 50,000 microliter, the treatment would be transfusion of concentrated platelets. Petechiae is actually formed when you have inadequate platelets. This is a significant symptom of low platelet count, among other things. So, bleeding disorder. Bleeding disorder is brought about by impaired liver function, the inability to synthesize clotting factors, which includes vitamin K deficiency, hepatitis, or cirrhosis. Liver disease can also prevent liver from producing bile, which is needed to absorb fat and vitamin K. Hemophilia includes several similar hereditary bleeding disorders, which is hemophilia A, hemophilia B, and hemophilia C. These are genetic bleeding disorders. Disseminated intravascular coagulation involves both widespread clotting and severe bleeding. This widespread clotting occurs inside an intact blood vessel, thereby blocking blood flow. Severe bleeding would then follow because residual blood is unable to clot because clotting factors are being depleted. These would occur in septicemia and incompatible blood transfusion or complications in pregnancy. This blood transfusion is actually going to be discussed in the lab. So I am going to end here because blood typing and blood transfusion is part of your blood. You are supposed to be doing this in in the lab. All right. So I'm going to end the discussion now.